Hello, my name is Jason Sinclair from the Animal Sciences Department at the University of Maryland. And our laboratory studies heme. Now, 1/3 of the world's population suffers from iron deficiency anemia. So most of the iron within the body is present in a cage called heme. Heme is mainly found as a component of hemoglobin within red blood cells. Now, hemoglobin is the protein that delivers oxygen throughout our body and in fact binds oxygen through its heme moiety. If a red blood cell does not produce enough heme, it cannot produce enough hemoglobin and thus resulting in anemia. So heme is critical, yet too much is toxic. Therefore, in order to better treat anemia, we have to understand how do red blood cells regulate their heme levels, and other cells for that matter. Now, this has actually been a very difficult process to study, and that's because cells make their own heme. It's very difficult to uncouple synthesis from other modes of regulation. However, our lab has previously discovered that the small soil-dwelling nematode, C. elegans, does not make its own heme, but in fact must acquire it from the environment by ingestion through its intestine. That makes them a great model organism for studying heme homeostasis because we can carefully manipulate how much access the heme has worm to. So using C. elegans as a model organism, my PhD work has led to a novel discovery, and that's that cells don't just regulate their own heme levels, but in fact can communicate to one another. And whole organs can communicate to one another to ensure that heme is adequately distributed throughout the entire animal. Of particular importance is the communication between the entry point of heme and the supply, the intestine, to the master regulator, the brain. And we have discovered two proteins that are critical for this communication. When the worm is in low heme conditions, it senses this and then upregulates a gene called HRG7, the protein product of which is secreted out of the intestine into the worm's body cavity and then remarkably taken up by the brain. In response, the brain will release a protein called double-1, which then signals back to the intestine. And we believe it's the coordinated action of these two proteins that allow the worm to maintain heme homeostasis on a systemic level. And now we believe that this work will have relevance to human biology because it's becoming more clear that in humans, like C. elegans, heme can move between cells. And so we hope that in the future, this work will lead to better treatment of anemia. Thank you.